the infection, and in finding the receptor, we'd know what the earliest sensors of the immune system were that became activated immediately after inoculation of microbes, possibly microbes of all time. And we might also learn more about sterile inflammation, which was really not understood at all at that time. I worked on this with conventional approaches, mostly biochemical approaches, some cDNA cloning approaches to try to find the LPS receptor, but none of those bore fruit. And by the year 1990, something was known about the pathways triggered by LPS. It was shown in that year by Wright and Ulevich that antibodies against a cell surface protein called CD14, which was of a leucine-rich motif variety, could block the LPS response. But CD14 had no cytoplasmic domain, and therefore there was a lot of mystery about how the signal got across the membrane. It was widely postulated that there must be a co-receptor to which LPS would be transferred or brought into apposition, and this would signal in some unknown way to provoke the response of myeloid cells like macrophages, which we had studied. Most people at that time, including ourselves, took TNF to be an important downstream effector of LPS function. And where the TNF gene was concerned, it was known that the transcription factor NF-kappa B had to translocate to the nucleus to drive transcription of the TNF gene. That led to the cytoplasmic accumulation of the TNF mRNA. It was blocked from translation in a certain way because of a UA-rich element that we identified in the 3' untranslated region of the messenger. But that blockade could be overcome by the signal from the receptor. And at this point, one would get translation of the messenger RNA, production and processing and secretion of the protein, and TNF would bind to its own receptor on many other cells to elicit inflammatory effects. In my mind at that time, the whole question in this scenario was, what is the identity of the receptor? And we decided to dedicate ourselves to finding that. Ultimately, we found it using a classical genetic approach. It happened that there were two substrains of mice, unrelated to one another, in which there was, in each case, a mutation that caused these strains to be refractory to LPS. The first of these to be identified was found in 1965 by Heppner and Weiss to survive challenge with almost any amount of LPS one would give, and the animals seemed completely nonplussed by it. These were C3H-HEJ mice, and there were other C3H substrains to which these animals were closely related that were normally sensitive to LPS. It was assumed that a mutation preventing LPS responses had become fixed in the population in the early 1960s. And by 1980, O'Brien and colleagues had shown that even though these animals were refractory to LPS, they were highly susceptible to gram-negative infections, particularly Salmonella typhimurium. Only 1 or 2 microbes was enough to kill these mice, whereas tens of thousands were needed to kill the normal LPS-sensitive strains, indicating that LPS sensing is important to mounting a defense against bacterial infection. By 1978, Coutinho and Mayo had found another mouse strain that just by chance was LPS refractory. These were C57 Black10 SCCR mice, and again, there were control strains that were perfectly normal in their response to LPS. In both the C3H-HEJ and the C57 Black10 SCCR strains, the defect was recessive, and Coutinho and Mayo crossed the 2 strains and found the offspring were refractory to LPS, indicating that they had allelic differences of the same locus, which came to be called the LPS locus. In those days, it was just beginning to be possible to positionally clone, that is, to clone by phenotype. And we decided to track down the mutation in these strains that prevented LPS sensing. One had first to map the location of the mutation, and we chose to do this with the C3H-HEJ strain. We set in order all of the markers on mouse chromosome 4 that were informative for a cross between C3H, HEJ, and SWR. And this is all the markers there were in those days. Looking at a total of more than 2,000 Mayoses and using also black 6 mice as a mapping partner, we finally got to a critical region between these two markers that we found in the meanwhile, B and 83.3, and we could say with confidence that the mutation must be between them. The next step was to clone all of the DNA across that part of the genome, then to fragment all of the pieces of cloned DNA, which occupied a total of 5.8 million base pairs, and to look for genes within them, because at that time, the arrangement of genes on the mouse chromosome was largely unknown, completely unknown for this interval. For the next 3 years, we identified pseudogenes in this area, not real genes at all, although they didn't come labeled as pseudogenes, and we had to do a lot of work to be sure that they weren't true candidates. And all of them were ruled out one after another. None had a mutation distinguishing C3H-HEJ from C3H-HEN, and I became quite concerned that we might be in the wrong region because of a mapping error. But one evening, I found in 3 separate clones of bacterial artificial chromosomes containing mammalian DNA in this region, pieces of a new gene that had no known function. This gene was called toll-like receptor 4 because of its similarity to the toll gene in the fruit fly, Drosophila melanogaster. And as I looked at it, it seemed to me to be an outstanding candidate. Like CD14, which I talked about a moment ago, this new candidate gene had leucine-rich repeats in its ectodomain, but it was a membrane-spanning receptor, just as we had hoped to find. It made sense to me that LPS might be transferred from one leucine-rich repeat receptor to the next, and whether that's really true or not, we don't know to this day. Another clue, though, was that on the cytoplasmic side of the receptor was a motif that was very similar to that seen in the interleukin-1 receptor. This receptor also had inflammatory characteristics. It bound a protein ligand, interleukin-1, and it was well known to activate NF-kappa B. It made good sense to think then that this receptor also would activate NF-kappa B and drive the expression of the TNF gene. There was also a story from the fruit fly, from the laboratory of Jules Hoffmann, that in flies, the namesake of the toll-like receptor family was important for protecting these animals against fungal infection. And I won't go through the signaling pathway of toll, but it's very similar, as we now know, to the signaling pathway of toll-like receptors. We dropped all of our work at that point, looking for new candidates, and sequenced the toll-like receptor 4 in C3H-HEJ and HEN mice, and we found a single base pair change, a transversion of C to A, that changed the coding sense from P, a proline, at a certain point in the cytoplasmic domain to a histidine. We didn't know for sure that this was a damaging mutation, but it looked like it probably would be. And it was the first example we had found. In the other strain, C57 Black 10 SCCR, we had a clearly damaging mutation. These mice didn't express the toll-like receptor mRNA at all, whereas the control strain did. And we also couldn't amplify any of the mRNA using reverse transcription and PCR. Eventually, we tracked down the mutation in that strain to a straight deletion that eliminated 74,724 base pairs. And here are the flanking sequences that lie upstream and downstream of the deletion. This was obviously a damaging mutation. The 2 genes were known to be allelic, and that to us proved the case that TLR4 was the critical signaling protein for detection of LPS. Going forward about a decade, it was verified crystallographically that toll-like receptor 4 did come into direct contact with LPS. There was also another subunit of the binding complex, a small protein called MD2, which bound most of the acyl chains of LPS and forced the formation of a dimer of dimers, which led to recruitment on the cytoplasmic side of adapter proteins that signaled forward into the cytosol. Other toll-like receptor paralogs have been found. We know of 10 in the human and 12 in mice, and all of them seem to bind molecules that trigger a response when bacteria are present. For example, TLR5 is activated by flagellin from both gram-negative and gram-positive microbes. Double-stranded RNA, usually of viral origin, binds to TLR3 and activates it. TLR2 together with TLR1 or TLR6 as partners in a heterodimer will bind bacterial lipopeptides and signal. And other nucleic acids are bound by TLR9, TLR7, and 8. Collectively, the toll-like receptors recognize most microbes that anyone might ever come in contact with. Much earlier than that, we had looked at the family of proteins. I've told you a little about the Drosophila story, and I've told you about mammals. Humans, like mice, depend on TLR4 to recognize gram-negative microbes and get into trouble if they can't signal by way of TLR4. The other TLRs have their stories in humans as well. What surprised me early on was that even plants had tear domain homologs, and in every case, those tear domains were part of plant disease resistance genes. Here, for example, in the flax plant, prevention of Melanpsoralini depends upon this particular tear domain protein in the cytosol. There are some plants, Arabidopsis and rice, for example, that have cell surface toll-like receptor facsimiles, which lack the cytoplasmic domain characteristic of toll-like receptors, but have other signaling mechanisms. I tell you this just to point out that we had found a really ancient innate immune signaling mechanism that arose probably even before the split between plants and animals. That work had taken us a period of 5 years, from 1993 to 1998, but I considered it was very worthwhile, deeply satisfying, and I decided that I would use forward genetics to find other phenotypes and track them down. These two phenotypes that I described, the phenotype of LPS resistance in HEJ and BLAC10 SCCR mice, those were mistakes of nature, but to make new phenotypes, we had to mutagenize the germline genome. And this was done with ethyl nitrosourea, and we began to do this in the year 2000. The idea being to find new immune phenotypes induced in the germline of mice, then track down the causative genes by positional cloning, and eventually establish a list of parts that were critical for immunity, at which point one can do experiments with the parts, find what associates with what, how signaling works, and eventually one could understand the machine as a whole. When we began this work at the Scripps Research Institute in La Jolla, to which I had moved in 2000, we made rather small pedigrees for the sake of frugality, really. Now, we would mutagenize all on a BLAC6 background, and mutations induced in the spermatogonia of male mice would be passed to male founders of the pedigree. We would breed these mice to a BLAC6 female and then cross them to their own daughters in order to make G3 mice, where there could be homozygosity for these mutations that were all heterozygous in the G1 founder. Typically, we would look at 6 such animals, reckoning that 6 mutations should be enough to capture, in at least one instance, each mutation present in the G1 progenitor. And if the phenotype was strong, we might very well see it and be able to map it. We looked at all phenotypes that we saw. Many of them were visible, but we were most interested in things that we had to measure immunologically in immune deficiency phenotypes. Failure to respond to mouse cytomegalovirus infection, for example, failure to make an antibody response, abnormalities detected in flow cytometric assays of the blood. In all, we tracked down about 200 phenotypes and solved them, and every phenotype resulted from a change in coding sense. That's very important, as you'll see as we go forward. One doesn't need to search in remote intergenic locations, for example, to find the vast majority of phenotype induced by EMU. In those days, we were a little bit naive about how we proceeded. We didn't look pedigree by pedigree. We considered every G3 mouse to be equivalent to every other one. And in this case, we were looking at TNF-alpha stimulated by challenging macrophages with lipopolysaccharide and looking for new exceptions to the rule that these mice would make a TNF response. Here was one such exception. It's one outlier out of several thousand mice. You might imagine that it's just some kind of a fluke, and we're dealing with some kind of false positive. But this one turned out to be true, though that was sometimes the case, that we just were chasing errors. When we bred this mouse and made a new generation of mice, we find they were all responsive to LPS and would make TNF-alpha. But if we back-crossed these animals to the founder, we got two populations, responders and non-responders. It was a clean separation, and we could track this down just as we had done before. In this case, we found a new adapter protein for both TLR4 and TLR3, which these days is known as TCAM1 or TRIF. That was one of the very first hits in our forward genetic gambit. I return to the fact that there are false positives, however, and false negatives were common as well. We were only looking for extreme phenotypes out of huge numbers of mice, and more subtle phenotypes would have been certainly ignored, even if we had taken a pedigree by pedigree approach, which we began to do. Let me show you an imaginary example. If we have a screen of some kind and we get a quantitative result from it, let's say the amount of TNF that's made, we might look at a single pedigree and find two exceptions from that pedigree, a large pedigree, relatively speaking, and we would look at them and we would say, this looks real to me, let's positionally clone it. But if the example had been a bit different and all the mice had been the same except for those two in their response, let's say these two gave a very subtle response defect, we would look at this pedigree and we would say, there's nothing here to be seen, let's go on to the next pedigree. But we could be wrong. If, on the other hand, we had a powerful tool of some kind, which was imaginary in those days, that said, somewhere in this pedigree there is a mutation for which these two mice are homozygous, the ones in green are heterozygous, and the ones in yellow have homozygosity for the reference allele, we would say this is a very unusual distribution. This mutation is probably the causative mutation. We better go try to clone it. These days, we do have this powerful tool. We developed this method of very sensitive positional cloning in 2013, and we now can recognize mutations as causative instantaneously, and we began at that point to positionally clone thousands of times more rapidly than we could in the old days. Let me explain exactly how it works. As ever, we mutagenize male mice on a black 6 background. We make G1 offspring that are carriers for mutations induced in the spermatogonia. But these days, all the G1 mice get whole exomes sequenced to identify all the mutations they might transmit to their G2 progeny and their G3 grandchildren. This is done using Illumina sequencing technology. We guessed in those days about how many coding changes there might be per pedigree. We now know that the average is very close to 60, but there's quite a lot of variation around 60. And we don't like to go looking at non-productive pedigrees, so we make a rule that we only consider those that have 30 or more mutations. If there are 30 or more mutations, we move forward and we order a set of primers to amplify all of those mutations in all generations of mice. This is done by AmpliSeq technology, and we, these days, make a large number of G2 daughters by harem breeding the G1 mouse, and we make an even greater number of G3 progeny, about 10 to 15 for the G2 generation, about 50 for the G3 generation. The next step is to genotype all of these mice for all of the mutations that were identified in the G1 progenitor. This is done using ion torrent technology, and therefore using two different methods, this one being particularly applicable for this purpose. We can be almost 100% confident of the reality of all of the mutations that were found by Illumina sequencing. Ion torrent technology applied to multiple pedigrees at once, in this case, 3 pedigrees at once, will show without ambiguity which mutations are homozygous, which are heterozygous, and which mutations are nullizygous, or simply reference allele homozygote, in every mouse in all of those 3 pedigrees. There's almost nothing that's ambiguous here. And only at that point are all of the mice subjected to various phenotypic screens. We do more than 100 different measurements on every mouse. Simple things like weight is determined, motor performance on a rotarod machine that tests the balance of the mice in a quantitative way. We look at innate immune performance, adaptive immune performance, antibody responses, including allergic type responses. We do flow cytometry on the blood of all mice to see if they have developmental or activation defects. And we look also at the bones of the mice, how they have grown, how dense they are, at blood pressure, heart rate, dextran sodium sulfate responses, also at the resistance of the mice to cancer, which I'll talk about a bit more later, addiction-related behavior, the structure of the eyes, circadian rhythms, innate fear responses, and autism-like behavior. And many of these screens are done in collaboration with other laboratories in our university. The computer, no longer a human observer directly, tells us when we've found a causative mutation, or at least a good candidate. The causative mutations are flagged and declared as candidates for causation based on statistical computation, and also on machine learning assessments that look at the primary mapping data for various qualities or features that I'll describe. The mutations are ultimately verified as causative or excluded by recreating them using CRISPR-Cas9 targeting in the germline of a non-mutagenized mouse, and then expanding the pedigree and retesting for phenotype. This assessment also is handled by the computer. We don't trust human beings who tend to fiddle with the absolute requirements for verification. The computer uses strict criteria to say, yes, this is verified, or no, it's excluded. It would help for you to see what this looks like. A pedigree that contains a mutation of interest is brought to our attention, and when we look at it, we first look at the Manhattan plot. We're looking here at a plot of the negative log of the p-value, testing the null hypothesis that each one of these mutations in the pedigree, which are arranged chromosome by chromosome, has nothing to do with the phenotype of interest, in this case, the T-independent B-cell response, that is the ability to make IgM antibodies to a specific kind of polymeric antigen called NP-Ficol. You see that there's one obvious outlier mutation from among all the mutations in the pedigree. This is the p.05 limit, and this is with Bonferroni correction because we're looking at so many mutations. We have one mutation that gives a p-value of better than 10 to the minus 9. And if you mouse over it, you see quickly this is the nicastrin gene. If you don't know what nicastrin is, you left click, and the computer already has collected quite a lot of information about that gene for you. You would learn quickly that this is a subunit of the gamma secretase complex. You would see where the mutation is that affects the protein. You could also look at a model of the gene and detect probable splicing errors if they exist, many other things, but I won't go through them all. I'll just point out that if you right-clicked on the Manhattan plot, you'd see the raw data. This is the phenotypic performance of homozygotes for the mutation, or VAR mice. These are heterozygotes for the mutation, or het mice. These are homozygotes for the reference allele, or ref mice. And here we have some added control wild-type mice that have no mutations induced by ENU. This, of course, wouldn't happen by chance alone. All of it was done blindly, and this is why it has such a strong score. In the same pedigree, there were mice that had light-colored fur, this being a normal C57 black 6 mouse. And these mutants could have been caused by, the phenotype could have been caused by another mutation in the pedigree. As it happened, also declared that the nicastrin mutation was causative of this phenotype. When we recreated that mutation, the knockout is not viable, so we had to make the exact same mutation to get viability. We saw that the T-independent B-cell response phenotype was just the same. And here we're looking at one of many alleles that we created, but this one duplicated the amino acid change. We also found yellow fur, and the computer reports to us with highlighting all of the phenotypes that we had declared before that did verify. And you can see that some of them really are probably verified as well, but they don't quite reach our strict p-value criteria. As you do this over time, you tend to hit the same genes again and again, sometimes even hundreds of times if the genes are very large. In this case, there were 7 pedigrees in which we had hit the diaphanous gene and we had seen the phenotype. The computer knows this, and it makes what we call a super pedigree in which we look at the immunological phenotype. Here we're looking at CD8 T cells that are central memory in their quality among all CD8 T cells. And the gene of interest is highlighted in red. It's shown as a red dot, where all the other genes, hundreds of them in this summation of pedigrees, are shown in blue. And we might have multiple alleles of some of the other genes too, but they're shown only in dark blue. The one we're analyzing is shown in red. If we click on that point, we find, yes, indeed, we have strong collective skewing from heterozygotes or reference allele homozygotes in this assortment of pedigrees. And the size of each point indicates the likelihood of damage caused by the mutation, large being likely null allele, intermediate being possibly damaging, and the small ones probably benign. This happens automatically then, day after day, and we can say that this collection of phenotypic outcomes gives this particular Manhattan plot, and this collection of phenotypic outcomes gives this Manhattan plot. And I won't go through what the assays are. I'm just trying to show you that it's a very common thing to have putative causative mutations these days. There are 50 more examples. There are 400 more examples, and you can probably barely see the data at this point. But the point I'm trying to hammer home is that now, as of a couple of days ago, we had made 22,320 declarations of phenotype ascribed to specific mutations, which means that we had completed 22,320 positional cloning projects, which used to take us years to complete. And this is the breakthrough technology that lets us really go into depth and treat the mouse rather like people could treat C. elegans in the past, pushing towards saturation and trying to understand many pathways. At this time, we have subjected more than 220,000 mutations to phenotypic analysis, and those mutations fell into 21,126 genes, and that is most of the genes that there are in the mouse, better than 90%. We've tested the null hypothesis, namely, this mutation does not affect the outcome of this phenotypic assay, more than 22 million times. And we've declared from all of that, as I told you a moment ago, that there are 22,320 instances of cause and effect, now emanating from 4,589 mutations in 2,275 genes. We can say then that about 1 per 1,000 tests of the null hypothesis leads to a declaration, ultimately. Where immunity and inflammation phenotypes are concerned, we now have nearly 3,000 causative mutations that fall into nearly 1,400 genes in our collection. And while some of the mutations were certainly known before, well over half of them were novel. That means we're really making progress. For our best screens, for example, for the flow cytometry screens, we've reached a state of 57.4% saturation. And that means that we not only screened the mutations, but did so with the mutant allele represented twice or more in the homozygous state, which we take to be sufficient to accurately assess a flow cytometry phenotype if it's really there. How do we know that they're really damaging or destructive? That's a long story, and maybe I'll answer it in the question and answer session. But we do have confidence that we've truly damaged or absolutely destroyed the genes with these alleles. There's more than just statistical computation to declaring causation, but there's no preconception. In other words, the computer knows nothing about the immune system. It does not rely on literature claims. It's only using the genetic data that we developed in our lab to make declarations. But it certainly can assess whether there are multiple alleles that cause the same phenotype. That gives support for causation by a weak mutation. There's also historical reliability of each assay that's taken into account, the damage probability of the mutation, the essentiality of a gene. If a gene is essential for life, then very often viable hypomorphic alleles have some effect on immunity. The quality of the data, are there extreme outliers? Is there overall a lot of variance? How many homozygotes were tested? What was the magnitude of the effect, and so on? All these things go into making the assessment. Co-segregation does as well. The experience with retargeting by CRISPR-Cas9 plays a part in assessment too. We humans try to bring those things into account, to weigh them properly, but we're not very good at that. We devised what we called the human algorithm, which went through and assigned our impression of how things should be weighted. And we subjected our tests of the human algorithm based on 487 targeted genes, where we reperformed the assays and then went back and asked how good is this algorithm using a receiver operating curve. And sure enough, it's not really like doing nothing, the diagonal curve would be. But if we look at sensitivity and specificity plots, we're only getting to a point of about 0.74 area under the curve. Again, good, but not really fantastic. We thought a computer that used real machine learning could do a lot better. And so we developed an artificial intelligence program that we call Candidate Explorer. And it utilized the human algorithm as one feature that it takes into account. Also, it looks at the essentiality of genes and at protein damage. And these are embedded machine learning tools that we also developed. And then there are 62 defined mapping data features that we feed to the program. And it gives us, in the end, a score between 0 and 1. Remember, this was how well the human algorithm did. Candidate Explorer does much better. The area under the curve there is 0.944. And I would say this is good enough for using in clinical diagnostics if it's not really a life or death situation. And maybe even if it is in some cases, if you can do no better. But here we're getting really quite close to the axes. We can take Candidate Explorer scores and put them into bins of 0.01 size. And we're looking now, by this time, at more than 5,000 reassessments based on CRISPR retargeting in a fresh mouse. And every point, therefore, has an average of about 50 measurements where we said, yes, it's verified, or no, it's not, to support it. If you have a low Candidate Explorer score, you have a very low likelihood of verifying the mutation. If you have a high CE score, you have an excellent chance of verifying the mutation. And one begins to think that perhaps this program, doing better than Human Observer as it can, is the way to go. And in fact, in this coming week, we are about to make CE completely autonomous in declaring mutations, whereafter human observers will no longer need to look at the data and decide whether a mutation is causative of phenotype. Candidate Explorer will do that. It will also continuously review our full database and at times withdraw mutations that were formerly thought to be causative on the basis of new alleles that have been found or new alleles that were formerly too weak to be considered causative in cases of co-localization. Co-segregating mutations are one of our major problems, working with small pedigrees as we do, and CE may rethink those as it gathers more data. When several million mutations have been tested, remember we're in the 227,000 range now, but we hope to go to millions. Ultimately, we should know all genes that can influence any measurement we've chosen to make in the Black6 mouse. The curve that I showed you a moment ago, the sigmoid curve, will become quite thin in the middle, and all genes and all candidates will be pushed toward the upper or lower bound of that curve. Already, we're beginning to see some approach to saturation. We started this to look at toll-like receptor signaling foremost, and we didn't continue it and make it one of our deepest screens. But we did find a total of 85 mutations in 39 genes, with multiple alleles shown in red lettering here, that influenced TLR signaling to the level of TNF biosynthesis. 16 out of these genes were new at the time we found them, and we have published on all of them at this point, I believe, and those are shown in blue boxes. At the same time, the field has become very active, and a lot of mechanistic work has gone into looking at these proteins. Not all of them are shown here, and some of the relationships I'm showing are fanciful, but some of them are real. We know about the midosome, for example, a kind of aggregate of adapter proteins that fix kinases and let them signal to drive transcription factor activation. And we know that parts of the signalosome fit together approximately as shown, and NF-kappa B as shown. So we are getting toward the extreme of knowing all parts of the puzzle and being able to reconstruct a watch. Certainly we're not there yet, but in our defense, I would say that the whole system is a lot more complicated than any watch that's ever been made. We've identified in the adaptive immune realm certain proteins that were not formerly known to be involved in immunity at all. And I think those of you who are seasoned immunologists might have heard of some of these by now. All of them cause quite profound immune disturbances and just weren't recognized based on any other mechanistic approaches that people have taken. There are actually hundreds of them, but the final part, really figuring out how they work, takes the longest, of course. And we're working on many others that aren't in this list. To this point, I've talked to you about how we've used this platform to make new genetic diseases of known cause that inform us immediately of things that are important in areas of biology that interest us. But it has perhaps occurred to some of you already that we could use the same platform to suppress disease. And if we can do that, finding modifier loci, then we have a potential drug target. And this target usually would be immediately applicable to humans with the equivalent disease state. We've begun to ask, can we suppress cancer? Can we suppress diabetes? Can we suppress arthritis? And in each case, we simply have to find a good facsimile of the human pathologic state in the mouse and mutagenize and see what the mouse would inform us of. The disease can be either environmentally induced, for example, put a mouse on a high-fat diet and see whether it fails to become obese, or it can be genetic in etiology. And I want to close with one really nice example. I won't go into great detail about it, but let's talk about the cancer problem. Probably there are humans out there who are quite resistant to cancer on the host side, not in terms of what oncogenes they might have that are mutated, but they simply are resistant to the progression of cancer. That's very hard to find using human genetics because most of us never develop cancer in our whole lifetimes. It's only the unlucky 1/3 of us who do. In mice, the situation is very different, of course. They're all inbred, they're genetically almost identical to one another until we introduce mutations with EMU, and we can look parcel by parcel, pedigree by pedigree, to see if we can find cancer resistance mutations. We chose to do this with the black 6 strain, as usual, using the B16F10 melanoma. And we give it an inoculum of 2 × 10⁵ cells per mouse, which is uniformly fatal to these mice under normal circumstances. We also give a checkpoint inhibitor, anti-PD-1 antibodies, which in humans will salvage something like 30% of people with melanoma. At least they'll have a kind of measurable response. Most people don't respond. No black 6 mice respond with this tumor to checkpoint inhibition under normal circumstances. The outcome of the screen is measured on days 13 and day 20 by measuring the volume of the tumor with calipers. And if mice don't grow a tumor at all, we re-challenge them 7 days later to make sure some kind of mistake hasn't been made. We do find phenotypes in this screen. Some of them are relatively weak, but verifiable. For example, we have this phenotype called starved, which we mapped automatically. These were the original data. And when we regenerated that mutation, it being a gene in which homozygous knockout is lethal, so we recreated the exact mutation. We found again, yes, there is some resistance to B16F10 melanoma here, but it's not that dramatic. All of the mice eventually would die of melanoma if we let them go long enough. On the other hand, there are very strong mutations. This one was called icebreaker to connote a kind of breakthrough, I guess. And it had a good p-value. And in the initial screen, we saw that there were 5 homozygotes that didn't grow any tumor at all. There were 3 heterozygotes that didn't grow a tumor also. And we re-challenged those mice that are shown by red dots. When re-challenged, 2 of these 3 did grow a tumor. And I don't know why this one didn't, because by now we've worked with this mutation very extensively. We know it's strictly recessive. Maybe a confusion of the ear tag, who knows? But these 5 didn't grow the tumor at all. So we were quite confident we had something real. And even before we made CRISPR-Cas9 retargeting, we grew a large pedigree from the survivors and had a further look. The icebreaker mutation affects a protein that's essential for life. Knockouts are lethal. On the other hand, heterozygotes for the icebreaker phenotype and homozygotes are quite viable. And if you cross icebreaker over plus, you get a normal Mendelian distribution of the 3 genotypes. But icebreaker over knockout is 100% lethal, just like knockout over knockout is lethal. And therefore, we know, at least where lethality is concerned, that this is a hypomorphic mutation. The protein is highly conserved between humans and mice, 99.3% identity, and the mutation affects an area of the protein that's absolutely conserved over a very long region. There was no previous awareness of resistance to cancer in mice with mutations affecting this protein. We wondered whether PD-1 antibody treatment was really necessary to rescue the mice. The answer is no, it's not. Looking here with a rather large number of mice, I think with about 15 mice per curve, you can see that the mutation is absolutely recessive. Here with the solid green curve, you see the performance of heterozygotes. If you take homozygotes, which are shown in red, and you don't give them a checkpoint inhibitor, that's the broken curve, these animals are very significantly resistant to B16 melanoma. On the other hand, if you do give them the checkpoint inhibitor, they're much more resistant. And that tells us something about how the mutation works. It's not only B16F10 melanomas that are inhibited in their growth by the icebreaker genotype. Here we're looking at MC38 colon carcinoma in the mouse. And we're looking at groups of 20 or 18 or 15 mice of the 3 genotypes. The icebreaker mice will grow the tumor. They support growth of the tumor till about 8 days. And after that, they rapidly resolve the tumor, rejecting it in 100% of cases. And now we've looked at hundreds of mice. There's no exception to that rule. We did go back and recreate the mutation with CRISPR-Cas9, but actually we were imperfect in doing so, and there were closely linked insertions or deletions that were in frame, and so these were viable alleles. They too have the same behavior. So now we could say we have 3 alleles of the icebreaker gene, and it does the same thing, protects against MC38 colon carcinoma, for example, does all of the things really that the original allele would do. Now, what can we do with that knowledge? By this time, the mechanism is known with great clarity to us. We know, for example, that this is mediated by CD8 T cells, but it's really a mutation that affects the thymic epithelium. We know that the mutation affects central tolerance to a specific peptide that's present in the gene that's been hit and modified. And this modification allows escape from the normal negative selection of the wild type peptide, allows the formation of TCRs, T cell receptors that don't normally exist and that recognize the wild type peptide where it's present on tumors that have the wild type peptide. This, interestingly, was a very rare event in our screen. We carried out screens on 32,000 mutations, and this was the only mutation that worked in that way, giving robust resistance to cancer based on a subtle change in central tolerance. We also know that tumor resistance can be transferred to wild-type mice if you transfer icebreaker T cells or CTLs, cytotoxic T lymphocytes that are tumor experienced. However, autoimmunity is not observed. There's no graft versus host disease, even when checkpoint inhibitors are co-administered, or even when you give the T cells to a RAD-deficient wild-type mouse, which lacks regulatory T cells as recipients. You can also transfer wild-type skin grafts onto icebreaker mice, and they're not rejected. Only tumors are rejected, indicating that a tumor-derived signal is probably quite critically important to get the process started. I will say again that humans also have the target peptide, and the question we are now asking is whether we can circumvent central tolerance in human cancer patients and expect comparable results targeting that peptide. I'll also mention that we have other strong mutants that prevent the growth of cancer in mice, and they work by totally different mechanisms, and there's early indication that they synergize strongly with the icebreaker mutation that I've shown you. So I'm optimistic, although we certainly have quite a ways to go. The story that I told you about the hunt for the LPS phenotype was carried out by Betsy Layton, Christoph van Uffel, Irina Smirnova, and foremost by Alexander Poltorak, who stuck through 5 years of bitter struggle to find that mutation, always knowing that he would in the end. These days, we have a larger group, and I will say that even this picture is out of date, and maybe that's a good thing, because the current picture would show us all wearing masks. Not present in the picture was Daohui Su, who wrote the Candidate Explorer program that's been quite critical to our recent work and prevents us from making many of the mistakes that we used to in evaluating mutations. With that, I'll thank you very much for your attention, and I'm very glad to answer questions.
Thank you very much indeed, Professor Beutler. Fascinating talk. It's far from my own field of science, but I just got a bit of a feel for the incredible amount of work that's gone into all of that. It's fascinating stuff. We have a good set of questions. We're not going to get through them all, but I will rush on to make sure we can cover a few. So Beginning with a question from Milena Malcharik. Professor Beutler, could you elaborate on the notion of sterile inflammation?
Yes, we certainly don't know as much about it as we would like to, but there are specific instances in which we see that toll-like receptors, for example, are involved in triggering it. This is true of the autoimmunity and accompanying inflammation that one sees in systemic lupus erythematosus, at least some kinds of it. We know that there's an influence of nucleic acid sensing toll-like receptors, for example, TLR7 in the mouse, or 7 and 8 in humans, TLR9 perhaps as well. You've got a kind of forward feedback where cells undergo lysis. This stimulates the release of DNA, and toll-like receptors are activated, and that helps to impel a B cell response directed against self tissues.
Okay, thank you, thank you. And Neda Farahi asks, how does LPS binding protein regulate TLR4 signaling? Is this a therapeutic target?
I would say yes, it is a therapeutic target, probably. Not too much is known. LBP doesn't give a very strong phenotype in our hands, but it is involved in either extracting or binding LPS in the plasma, probably escorting it in some way to CD14, which in turn is involved somehow in coordinating the TLR4 response. But those are not part of the watch that I showed you. We don't have good crystallographic evidence of association between CD14 and TLR4 to this day, but it might happen. It's a good thing to work on, and a good target, certainly.
Okay, thank you. Another question from Mohammed Iqbal. Wonderful talk, amazing work. I must say there's many comments like that. Can you give a comment about what factors make some people respond unfavorably to LPS, leading to septic shock, but not in some others? Is that down to the genetic mutation, similar to what you found with the LPS resistant mice, or is it because TLR4 gene has multiple isoforms?
It may have sometimes to do with the signaling itself, but my feeling is more often it doesn't. We did at one time in the old days have a screen for a sensitivity to the lethal effect of LPS, and we expected we might find new components of the TLR apparatus in that screen, but we also found other things. We found an ATP-sensitive potassium channel, for example, wherein mutation was quite viable, but if you gave a mouse LPS, it would die within a few minutes, actually. It would die probably a cardiac death. We know that this receptor is in the coronary arteries of mice and causes vasoconstriction, or let's say causes vasodilation in the wild type form. And the mouse can't make the right accommodation to survive LPS challenge, even very early on. Also will die of MCMV infection. And I think there are a lot of physiologic accommodations that go into surviving severe infection. Probably that is where much remains to be found.
Okay, we're running short of time, but I will try another question. Daniel Gale asks, well, he begins with amazing work, thank you. As you approach saturation using an exome-based approach, can you scale up to a whole genome sequencing-based screen? Can you integrate human biobank data sets into Candidate Explorer?
We can certainly look for correlations with human phenotype genome sets of data where they exist. We would like actually to look at whole genome sequencing, but it's still too expensive to do it that way. And I think that may change. It's certainly not a computational problem anymore to do it that way. And we likely will go in that direction. There's one other little quick comment, and that is ENU is a point mutagen. And if we used some other kind of mutagen, chlorambucil or X-rays or something else that's much less convenient to use, those tear out large chunks of DNA, and we might see that there's a much bigger intergenic contribution than we're seeing with a point mutagen.
Okay. Right, I see we're up to time really, and that there are a few more questions, but I'm sure we could go all evening. I will thank you very much again, Professor Beutler, for giving up your time and presenting to us on your work. It's absolutely fascinating stuff. Thank you.
Well, it's a pleasure. I hope I can visit in person someday.
Absolutely. I was going to say that the least we could do is offer you a nice dinner in college if you're in Cambridge. So next time you're in the UK, please do let us know.
Okay.
And I will say thank you to our audience. We're up above 100 people joining in and asking questions, and apologies to those who didn't get their questions answered. This event has been recorded, so there will be access to it and you can look back at it. I'll now pass back over to Natanya.
Yes, I just wanted to say thank you so much, Professor Beutler, for a very interesting presentation. I'm very excited to see where the field is going to go next. And thank you to Professor Barber for hosting this event and to the audience for coming. I hope you all have a lovely evening.
Thank you all.
Thank you very much.
Good night.